Hello and welcome. Thanks for clicking on this one. So this week we'll be discussing possible treatments for Alzheimer's disease, and today's article, Advanced Nanomaterials for Modulating Alzheimer's-Related Amyloid Aggregation. So we're going to be looking at nanotechnology essentially as a way to treat Alzheimer's disease. This article was published 2022 November. Shao are the authors. You can kind of pause the video here and kind of write everything up, and if you want to go check it out, it's free. It's in PubMed. So here's the abstract. Finally, the challenges faced by nanomaterials with different dimensions in AD-related amyloid aggregate modulations are expounded, and the prospects of nanomaterials are proposed. So the abstract is essentially talking about what this paper will be, as you know, what abstracts essentially do. And the main takeaway here is that basically, Alzheimer's disease exerts a lot of economic pressure on families, and we need to find cost-effective ways to treat this disease. And here, the authors are propositioning nanotechnology, specific types of nanotechnologies that can be used as drivers to take medicine up to the brain and, essentially treat or remove the amyloid plaques, as well as some way to scaffold the remaining neurons so it can help the brain heal, essentially by kind of like the overwhelming reactive oxygen species that are released from the amyloid plaques, as well as when microglias are activated, can kind of overwhelm the entire system as well. So when you have these drivers that are coming in with also them having antioxidants equipped on them, they can also help relieve the amount of antioxidant species that are there by exerting the antioxidant action. So they're able to. So in summary, these nanotechnologies are able to be drivers, bringing medicine into the brain that can help essentially obliterate the amyloid plaques. And on top of that, the nanotechnology has the capacity to also be antioxidative, in which case on top of it actually chelating ions, which that means is it's able to capture ions that are also charged, which can also be oxidative in nature. So this can act with a reactive oxygen species. So on top of it being able to capture those haywire ions like copper 2, for example, it's also able to capture the widespread oxidative species that are there and perform potent antioxidative action, relieving some of the damage that can come from the release of all that. So that's basically it. From here, we're just gonna switch gears and take it to the PowerPoint here that I have set up from Canva. Great website, by the way. So advanced materials for modulating Alzheimer's related amyloid aggregate. So what's Alzheimer's disease? So we know what Alzheimer's, if you haven't clicked on any or seen any of the other videos, I'll explain here what Alzheimer's disease is basically. The main cause that we know now is the amyloid hypothesis, whereby you have amyloid aggregates. Amyloid is a type of protein that is released from cell membranes. Usually in normal action, it's released and then eventually it's taken away by CSF. But in Alzheimer's disease, what happens is the person who has it will release a mutated version of this protein that is able to not be cleared out. It's insoluble, and this protein monomer that's mutated can link up with other protein monomers and essentially cause clumps and essentially become aggregates, oligomers, and that affects the signaling of most of the neurons, and neuronal cell death happens. And then you have the addition of also some neurons also exhibit tau hypophosphorylation. Tau is a type of protein that is inside cytoskeletons of axons and neurons, and what happens is the tau proteins become mutated as well, and then they cause clumps, just like the way the amyloid better fixed together. The tau they cause clumps within the axons, and that causes them to, essentially prevent the neuron from being able to go from its cell body to the terminals, and then essentially it dies off because it needs the constant stimulation from the other neurons from its terminals in order to stay alive. So the neuron dies from the inside and from the outside. It's a double whammy approach. So it's like when it comes to treating this, there's multiple things that are happening that have to be treated. You can't just address one thing and forget about the other thing. So that's why it seems like treating this is kind of taking a whole other way in which people are just thinking outside the box, and nanotechnology has come to be one of those thoughts. And so let's just explore more about what it is. So let me just kind of highlight what I'm trying to talk about, what type of technology we're going to be talking about in this paper. So There's zero dimension, one dimension, two dimension. So the main thing that you need to just take away from the zero dimensions, these are the smallest ones out of all these. They tend to be just like spherical in shape, especially for the purpose of this article. Just imagine most of them are just spheres. Either they're big spheres or they're small spheres. And then we have one dimensional. One dimensional, usually they're tubes. It's just kind of one plane essentially. And then two dimensional, those are usually sheets. So think of them as that essentially. So that's just it. The paper is not going to be talking about three dimensional, so we don't need to know. We don't need to worry about this for now. But these are the main kind of nanotechnologies that we're going to be discussing in this paper. All right, so let's begin here. So yeah, so one-dimensional nanomaterials review. So with this type of technology, the one-dimensionals are usually like tubes, and they're able to, for the most part, kind of cross the blood-brain barrier, and they are, especially when they're hydroxylated, they're able to bind with the amyloid beta oligomers in more affinity, and this kind of helps the CSF kind of clear out most of these. And when it's able to do that, at least it has it's biodegradable, so it can stay within the neuronal stream for a long time. It's able to cross it very easily, and it's very cost effectively. So it's also a pretty good driver, and it seems to kind of clear the amyloid for a long time, and it can do the action. But the main takeaway away from this is it might if the fact that it's biodegradable means that it's not going to be staying for too long. So we have two-dimensional nanomaterials overview. What we know from them is that they have a larger surface area. So as you can remember, these are the sheets. They have a larger surface area, so they're able to kind of stop the oligomers from forming, as well as when they have formed, they're able to kind of separate them because they have a high affinity for the hydrophobic region of these. So those amyloid plaques, they tend to clump together in a very specific way, whereby you have the hydrophobic heads tend to be inside, because you have to think of the lumen of the brain as kind of like liquidy. So the hydrophilic ends will be on the outside, hydrophobic on the inside. This two-dimensional nanomaterial is very hydrophobic, so it's able to clump up together or kind of disrupt the salt bridge of those hydrophobic parts of the amyloid pellic, and so that in that way it's able to unravel it. And when it unravels, it just kind of like loops around the sheets of these two-dimensional nanomaterials. So graphene oxide is an example of a one-dimensional uh, nanomaterial. So this is known to absorb amyloid monomers. It redirects amyloid beta and sends pathways larger. Graphene oxide sheets show better efficacy, prevents better sheet formation. When the amyloid plaque forms from, it usually forms like a helical structure at first, and then it becomes beta sheets. Beta sheets are just basically like the sheets that we showed earlier. And when it forms those sheets, that's when it becomes its most lethal, because it can cause more damage. It's usually longer, and it has more widespread action, and capacity to cause additional oxidative stress to the entire neuronal space. So you want to prevent this from happening, and this graphene oxide is very good at doing that. These two are from the two-dimensional uh, two-dimensional nano materials. We have black phosphorus, graphite, carbon nitride, and as you can see, it's activated by irradiation. Irradiation is basically when you shine a light towards this thing. When you shine a light towards this substance when it's at a far distance, and when it's basically inside usually it's a mouse. When it's inside the mouse's brain, and when you shine a light to it, it's able to it's able to change its shape based on the type of structure that it is. So they're just small enough that light is able to basically influence it. So this enhances reactive oxygen species reduction, oligomer removal. It's photocatalytic, biocompatible, and very low toxicity. Black phosphorus, it captures the copper ions that I was talking about. So copper is basically something that is released from the widespread danger that's caused by the amyloid plaque, and the fact that you can see it's charged there. It's very oxidative, so it's going to want to share those electrons with something else, which leaches other. It doesn't matter what it chooses. It could be another free electron, or it could be a cell membrane electron, and that can disrupt another neuron, and that can cause additional problems. So black phosphorus is very good at adapting it or chelating the step of ions. It prevents reactive oxygen species generation, which is what the copper 2 plus will do. Enables photodynamic therapy, which means light can be used to adjust it well inside the body, and it's able to cross blood-brain barrier without a problem. So zero-dimensional. These are usually very small, they're very spherical, biocompatible, ideal for detection, drug delivery, and treatment. So, quantum confinement effect imparts unique properties. Examples include gold nanoparticles, metal oxide nanoparticles, and quantum dots. So, quantum dots are usually used for detection of Alzheimer's disease. It's very common. And the key findings are: this is one of the type of just a type of zero-dimensional nanomaterial that was used, and it's basically gold that is um, made into a nanoparticle. And it's usually, the fact is that usually the ones that are negative charged are able to protect neurons from amyloid beta damage. Black phosphorus quantum dots, they're able to stabilize alpha helix and reduce amyloid aggregates. Selenium quantum dots are able to penetrate blood-brain barrier, scavenge radicals, inhibit amyloid beta aggregation. And we know quantum dots offer targeted delivery detection and neuroprotective effects. So this is just something that you have to take into account when you are going to be looking at what might be working in the research. So if you want to know what's been shown to actually consistently work in many of these researches, you can go in and look at these type of things. So gold nanoparticles is the first thing. The second thing is black phosphorus quantum dots. Third is selenium quantum dots. And quantum dots essentially are just very good at detecting and treating as well, as you can see here, based on this paper. The current challenges and future directions. Most studies are preclinical, so human trials have not been done. So most of these studies were animal trials, essentially when they were testing um, these nanomaterials inside them. But in most cases, when you look at mouse, for example, which is kind of usually the main animal that's used, they have very similar chemistry to human brains, so it can be transferable to a certain effect. But it's very complex because you can't really compare human brains to mice, because human brains are quite larger, obviously. So their affinity and durability of nanomaterials require enhancement. Need improved blood-brain barrier penetration methods. Targeting specificity is needed. Long-term safety and biocompatibility is also needs to be established, and we have to have better ways of translating in vitro to in vivo in clinical applications as well. Yeah, so the main conclusions are, different dimensional nanomaterials are unique complementary advantage for Alzheimer's treatment. One-dimensional nanoparticles provide high surface area and optical properties for aggregation inhibition. So, as it says, one-dimensional is not the best. Two-dimensional nanoparticles offer larger surface areas with multi-mechanism therapeutic effects. And zero-dimensional nanoparticles enable target delivery, detection, and protective effects. Combining nanomaterials could maximize therapeutic benefit, and the future focus on clinical translation human trials remain essential. All right, yeah, thanks for watching. You have a good one.